Yo, 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 welcome to the Unit 3 exam overview. Disclaimer, this isn't everything on the exam, but it helps you frame your ideas for concepts on the exam. I'm going to go over it. Again, everything on cellular energetics except for enzymes. So let's get started. First off, we have to talk about energy. So free energy is the energy that is available to do work. And that's essentially what you need to frame your mind around. Things in a cell occurs, and there's this concept of energy that helps the cell do all that work. We need to frame it around these two big ideas of metabolism. The first idea is catabolism. Catabolism is breaking down molecules into simpler molecules, and in that process, it releases energy. One way that I remember catabolism is to say my ABCs. So since this starts with the C, I'm going to go back to A. And that essentially is breaking down the alphabet. So breaking down molecules, breaking down the alphabets. So we need to tie free energy to it. So this is very equivalent to a negative delta G. That means energy is being released. So releases energy. And this has a term, exergonic. The big example for an exergonic or catabolic reaction is cellular respiration as we are breaking down glucose. Then we have the term anabolism. So again, my ABCs, but I'm going up my alphabet. So I'm building my alphabet. So I'm building molecules. So building. Building things requires energy. We can tie this again to free energy. This time, positive delta G, which means requires energy. And to describe it with the reaction, it's saying that it's going to be endergonic reactions. It is a reaction that requires energy to build something. And the big one is photosynthesis. We are building glucose from sunlight. Let's talk photosynthesis. So, photosynthesis. You guessed it. It is a redox reaction. And the overall reaction is taking light energy and converting it into chemical energy. So let's see the equation. The equation is, and I always start off with 6H2O plus 6CO2. Again, doesn't work without that light to create glucose. And 6 oxygen molecules. Let's label what is being reduced and oxidized. So I'm going to underline my first one. When I am trying to determine this, I always look for the change in H. So I see that there's a hydrogen in my water. Once I go over this right here, arrow, I'm going to look for a molecule that doesn't have H, which looks like to be oxygen. So Leo says Ger guides me here. Losing electrons is oxidation. So I know my water is being oxidized. And then for the gaining electrons is reduction. And so I'm going to look for the same change. That doesn't have an H. This one has an H. So going from no H to H is reduction, or it's got reduced. So the H represents the electron. So that's an easy way to quickly determine what is oxidized and what is not oxidized, or what is reduced. So I am going to be summarizing the photosynthesis process through arrows, just like we did in class today. So photosynthesis has two parts. The first one is the light reactions. The light reactions occur in the thylakoid membrane. We need to know locations here. And then I'm writing it out in terms of the arrows that you saw. So light stimulates or is absorbed by photosystem 2. This excites the electrons in chlorophyll A for it to travel to the ETC or the electron transport chain. Now, this process, y'all, leaves chlorophyll A empty, so photosystem 2 enzymes drags water, splits it into oxygen and hydrogen ions, and then the electrons go to photosystem 2. Now the electrons are very high energy at this part of our process, and so the electrons move through the electron transport chain to arrive at photosystem 1. When they get there, they have lost all of that high energy from the sunlight. That energy, though, is then used to move hydrogen into the lumen, which would contain high amounts of hydrogen ions. And this one has low hydrogen ions, okay, in the stroma. So now, photosystem 1 collects light, okay? The electrons got excited again, and now it is taken by an enzyme called NADP plus reductase. This reductase, y'all, puts the electrons onto NADP plus, turning it into NADPH. And from there, it goes to the Calvin cycle. So all of this is happening. Guess what, y'all? This part is also happening. High hydrogen concentration from the lumen diffuses through ATP synthase to create ATP, which also goes to the Calvin cycle. So we have completed the light reactions. Now I'm going to be covering the Calvin cycle. So part 2 of photosynthesis, Calvin cycle. Calvin cycle again occurs in the stroma. We will start off with CO2. CO2 combines with RUBP with the help of rubisco, an enzyme. This creates 3-PGA, a low energy 3-carbon molecule. That low energy 3-carbon molecule will then be energized in the next step to turn into G3, and I'm going to write that better, G3P. It gets energized by ATP from the light reactions. It gets energized by NADPH from the light reactions. So G3P, one of it goes to making glucose. The other, again, is used to, whoops, I thought I could connect that, my bad. The other 5 is used to regenerate RUBP by utilizing ATP. So let's label the major steps. First one, we have carbon fixation. When you hear that word carbon fixation, it simply means that we're taking CO2 and incorporating it into an organic molecule. The next step is reduction. Reduction just simply attaches the high energy electrons and phosphate to G3P, making it high energy. And the cycle is complete when RUBP is regenerated in the step called regeneration. Okay, that's the Calvin cycle, and that is photosynthesis. So we now are going to talk about cellular respiration. So I'm going to write this one in red. Cellular respiration. So what's the big picture of cellular respiration? You guessed it, it is a redox reaction. And again, it takes a chemical energy and converts it into a chemical energy. Whoa, glucose all the way down to ATP. Let's take a look at the equation. We have our big player, glucose. It is being reacting with 6O2 to create 6CO2 plus 6H2O plus a whopping 38 ATP. Now we're going to label what is being reduced oxidized. Well, my glucose looks like it is being oxidized. When you see that word oxidized, I also think of it as breaking down. And you can see that there. Glucose is broken down all the way to a single carbon dioxide. Okay, and then now on the other side, I'm going to use some sparkling colors here, and it is being reduced. Oxygen is reduced to water. Still need to know the redox, y'all, of these reactants. There you go. I'm going to draw the linear structure of the cellular respiration process. So starting off with 1, we have glycolysis. Glycolysis occurs in the cytoplasm or cytosol. We take glucose, okay, and then we're going to run cellular respiration through terms of glucose, single glucose. So let's follow that. Glucose is broken down into 2 pyruvates. And I'm always going to trace the number of carbon out for you so you can see. Now the next one is 2 ATP. That is generated by substrate level phosphorylation. Then 2 NADH, which goes to the ETC. Second step, we have pyruvate moving into the mitochondria, and it is broken down. So pyruvate oxidation. This happens in the matrix. Now, 2 pyruvates again breaks down to 2 acetyl-CoA. So what does it look like? We have a 3 carbon now to a 2 carbon. What's released along the way? 2 CO2s and 2 NADH, which goes to the ETC. Finally, our acetyl-CoA is going to be completely oxidized to CO2 in the next step. This is called the Krebs cycle. Happens in the matrix. And it has another name. It's also called the citric acid cycle, and you need to know that. What happens here? 2-acetyl-CoA broken down into 4 products, y'all. Okay, 4 CO2, so it's completely oxidized now. It releases 2 ATP through substrate level phosphorylation. So I'm just going to simply write down substrate level because you already know that. And then it releases 2 FADH2 and 6 NADH. So this again, y'all, it goes to the ETC. Well, we have our final step, y'all. A lot of stuff to write down here. Oxidative phosphorylation. It happens in the inner membrane. Now, we have, again, 2 parts to it. The first part, and I'm going to draw a bullet point, is ETC, or the electron transport chain. So I'm going to describe it. Electrons move to build up a proton gradient. And again, something is causing the electrons to move. Once NADH and FADH2 drops them off. That is oxygen. Oxygen accepts the electron to form water. That happens at the ETC. So this again, y'all, is the first step. The second step is chemiosmosis. Chemiosmosis. So chemiosmosis, y'all, again, has a required hydrogen ion concentration. So hydrogen ions diffuse from the intermembrane space to the matrix through the ATP synthase to create 34 ATP. And again, with that said, y'all, we have completed cellular respiration. Everything is counted for. Now let's talk about fermentation. Fermentation, again, is one of the pathways of anaerobic respiration, or pathway, anaerobic pathway, let's just say. So, what does fermentation do? The point or goal of fermentation is to regenerate NAD+. So let's take a look at that. Glucose, again, is undergoing glycolysis, so it breaks down to pyruvate. And then that pyruvate has 2 options. It has this option here with oxygen. It goes or undergoes oxidative phosphorylation inside the mitochondria. Obviously, all the other steps are there as well, such as pyruvate oxidation and Krebs cycle. It produces a lot of ATP with oxygen. That's the point. And then there's the option of no oxygen, where it undergoes a process called fermentation. With fermentation, y'all, it's still in the cytoplasm, and there's 2 types. The first type is called alcohol fermentation, and it produces ethanol, which is a type of alcohol, and NAD+. Along the way, with that conversion of pyruvate to ethanol, CO2 is released. Second type, which happens in our human cells, is lactic acid fermentation. The product that's being produced would be lactate and NAD+. Okay. Why is that NAD+ important? In glycolysis, we know 2 ATP is produced, and then also 2 NADH are produced. But again, to drive that NADH production, NAD+ must be present. So the NAD+ here feeds into glycolysis over and over again as long as there's the source of glucose available to continue the 2 ATP production. So we again can say glycolysis produces 2 ATP. Fermentation allows glycolysis to run over and over and over again. So again, fermentation is a pathway that allows glycolysis to occur over and over and over again. Glycolysis occurs over and over again as long as there is glucose. Okay, y'all, with that said, that's the framework of what you guys need to know. Have a good one.